Activation of GQ in endothelial cells lining blood vessels leads to NO release and blood vessel dilation. And in these pictures, in A, you have a cross-section of a blood vessel where you have the endothelial cells in the interior, where, as we said before, the apical side is facing the lumen of the blood vessel, a cavity, and the basal side is binding to the basal lamina or the basement membrane. On the other side of the basement membrane or basal lamina, you have the smooth muscle cells, and these are a different cell type of different differentiation status. Now, the smooth muscle cells, when they contract, that will lead to constriction of the blood vessel and an increase in blood pressure. Whereas when they relax, this lumen area will get larger, and that will lead to blood vessel dilation, and that will lead to a decrease in blood pressure. Now, this signaling pathway involves two cells, where the initial signaling molecule, acetylcholine, is going to interact with its receptor on the endothelial cell. So that's the cells that are actually have their apical side facing the lumen of the blood vessel. And the activation of the signal transduction pathway in the endothelial cells by acetylcholine will lead eventually to NO release by the endothelial cell. Now, NO is a small nonpolar molecule, and it will simply diffuse out of the cell, as small nonpolar molecules do, and go down its concentration gradient. Since NO is being made in the endothelial cell, you will have a higher concentration of NO in the endothelial cell and less outside, and of course, none in the smooth muscle cell. So NO will diffuse out, and it will diffuse in to the smooth muscle cell. And there, its actions will lead to smooth muscle relaxation. So here, we have in the blue rectangle the endothelial cells, or at least the pathway in the endothelial cells. Acetylcholine is the signaling molecule, which will activate the G-protein coupled receptor. The G-protein coupled receptor will act as a guanine exchange factor and activate GQ, its G-protein. The G-beta gamma subunit of GQ will go on to activate PLC-beta, just as the PKC pathway. PLC-beta activation will lead to the activation of IP3, remembering that IP3 is made from PIP2, the hydrolyzation of PIP2 to DAG and IP3 by PLC-beta, the enzyme that's involved in that hydrolyzation process. Now, we're not interested in what DAG will do, but we're interested in what IP3 will do in this pathway. IP3 will lead to activation of IP3 receptors on the ER, and these are calcium channels that will open, and that will lead to the release of calcium, or the activation of calcium, remembering the activation of any second messenger is a concentration increase of that second messenger within the cytosol. Calcium activation leads to the activation of NO synthase, an enzyme which will take arginine and produce NO. Now, NO will diffuse out of the endothelial cell into the smooth muscle cell. NO will go on to activate guanylyl cyclase, and guanylyl cyclase will go on to catalyze the reaction of converting GTP to cyclic GMP. Cyclic GMP is a second messenger. Again, the activation of cyclic GMP is a concentration increase in cyclic GMP within the cytosol because cyclic GMP is a second messenger. Cyclic GMP will go on to activate proteins that are involved in smooth muscle relaxation. And once you have smooth muscle relaxation, you will get dilation of the blood vessels, and that will lead to a lowering of blood pressure. Molecular mechanism for smelling, the olfactory pathway. The cilia of olfactory neurons contain olfactory receptors, which are G-protein coupled receptors. And here we have a picture of an olfactory neuron, and you can see that you have this structures as part of the cell that look like dendrites on a neuron that are sticking out from the soma of the olfactory neuron. And these are called the modified cilia. And the G-protein coupled receptors that are involved for smelling are on those cilia. The olfactory neuron synapse with neurons in the CNS, the central nervous system, and you can see that the axon goes off, and that's where an action potential will travel, and then the axon terminals of that axon will synapse with neurons in the central nervous system. So we have the signaling molecule. This is the molecule that you'll be smelling, binding to its G-protein coupled receptor on those modified cilia. These are the olfactory receptors, and this will lead to the activation of G-olf, a G-protein. The trimeric G protein, the G-alpha subunit, will activate the next step. So the G-alpha subunit of G-olf goes on to activate N-enol cyclase, very similar to the pKa pathway. Then N-enol cyclase will go on to activate cyclic AMP, and again, cyclic AMP is a second messenger. The activation of a second messenger is an increase in cytosolic concentration of that second messenger. Cyclic AMP goes on to activate cyclic AMP activated cation channels. These are ligand-gated cation channels, where the ligand binding pockets are cytosolic instead of being extracellular. As cyclic AMP concentrations go up, those ligand binding pockets become occupied with cyclic AMP and they will go to the open position. As we know from the previous section, if you open up a cation channel on the cell membrane, mostly sodium will come across. And if you bring positive charge from extracellular to intracellular, you will go ahead and depolarize the membrane. And the depolarization of the membrane, this is similar to an EPSP in the synaptic connections, because now those modified cilia, which kind of act like dendrites, are becoming depolarized. And if there's sufficient depolarization, that will lead to an action potential being generated. And that action potential will go on to travel down the axon of those olfactory neurons and depolarize the axonic terminals, the axon bulbs, the presynaptic membrane, which will lead to neurotransmitter release.